Hello, welcome to another screencast. I'll be discussing an introduction to the regulation of gene expression. And so we're going to just be reviewing, spending some time reviewing here and then using that review to go ahead. So we're going to review what we mean by negative feedback and reviewing allosteric inhibition and activation. So this is of enzymes because enzymes are involved in this process. And then describe and explain here how the negative feedback can alter the expression of a gene kind of in general, and then we're going to look in future screencasts about specific examples of this. Okay, so reviewing negative feedback, remembering that negative feedback, I prefer to think of it more kind of graphically again, where depending on what you're looking at here, generally over time, the concentrations or the values are going to sort of be fluctuating, right? So you have this sine curve, you have generally an upper limit, you generally have a lower limit, and so whatever you're measuring over here exists within a range, right? And so this range is kind of then what we define as being normal, right? So this could be rates, this could be concentrations, this could be values, right, et cetera, temperature, whatever. You're going to have this, this fluctuation from low to high. Right? And if you go beyond that, or as you approach the limit here, something will happen in order to bring that value to come back down. This could be population size, right, predator-prey relationships, et cetera. So something goes below that limit, something happens to correct, and you bring it back into that range. If you stay up above this limit or below this limit for too long, then the cell dies, the organism dies, the population crashes, etc. So we see this very similar thing here. So the idea of negative feedback is that as you approach these limits, something is happening to drive you back away from negative, the direction that you're going, right? So if body temperature is increasing, something happens negative to go away from that increased temperature and come back down. And then likewise, if your body temperature is too low, then something will happen to drive you back away from that and get the body temperature to come back up, right? Population size, enzyme, rates of reaction also. All these things can apply to this general curve here. So the way this happens, the feedback, so this is the negative kind of part of that, staying away from the limits within a range. The feedback part is that typically the end product here will feed back to the beginning and provide information. So this feedback is info, right? So this is either like temperature, this is some sort of stimulus, right, which is going to cause that feedback to then, that negative reaction to come back. So if you have a concentration being too high, the concentration here can inhibit this reaction right here. Therefore, you don't get B, C, D, and your concentration then drops down. Likewise, if the concentration is too low, that then signals, this is still negative feedback, because it's then going to enhance this reaction, so this reaction happens faster, and you're going to get more of this D product being built up. Right? And then too much is made, and that negatively feeds back, and then back you go there. So there's the negative feedback review. Competitive and allosteric inhibitors regulating enzyme activity. We talked about that most enzymes are multiple parts there, and we have this allosteric site that is not the active site. Right? So this allosteric site is the regulatory spot on the enzyme, and these enzymes have this active form or an inactive form. We talked about if you bind an activator to that allosteric site. Notice this is the allosteric site, not the active site. The activator binds there, you get this is stabilized in an active conformation. If you bind an inhibitor to that allosteric site, then you get this inactive conformation. Right? And most enzymes can go back and forth between these two. The concentrations of activators and the concentrations of the inhibitor, so the concentration of the activator and the concentration of the inhibitor, will play a role in how frequently you have this active conformation or how frequently you have this inactive conformation. So if you have lots of inhibitors built up there, you're going to decrease the rate of that enzyme reacting, right? Because you're going to be shutting off this enzyme, right? Not active or inactive, right, conformation as compared to the active conformation. So on the diagram on the right over here, we had this example earlier where we were making this product down here, this isoleucine thing, and the concentrations of isoleucine actually act as an allosteric inhibitor. So that allosteric inhibitor binds to the allosteric site, and then that changes this conformation right here of the active site. Notice that the active site is changed back over here. So we change this active site. It's no longer active, and so threonine can't bind to the active site there. If you can't bind threonine, you can't make this intermediate A, you don't make this intermediate B, and so on and so forth. And then this product here is going to go down, of course, assuming that it's being used. Right? And so as the concentrations of isoleucine would go down, then you would not have this interaction right here happening as frequently, which means you would have more of an active conformation to fit threonine. So you fit threonine, you can then make intermediate A, make intermediate B, and so on and so forth, and then your isoleucine concentrations come back up. Right? Again, negative feedback, keeping the concentration of isoleucine in this example in range. Okay, how does all this apply to then gene expression? Well, if we take that isoleucine example, right, we had this, we have some sort of like precursor molecule that looks like this, and then we want to eventually make it into this thing down here called tryptophan. Right? This is just a specific example. And let's say that in this metabolic pathway, there are 1, 2, 3, 4, 5 steps in that metabolic pathway to get tryptophan. And each of those steps is mediated by an enzyme. Right? So in this case, the tryptophan itself will act as a negative feedback molecule and feedback here and inhibit enzyme number 1. Right? So this probably acts as an allosteric inhibitor, much as we've seen in that previous slide. Right? But as it turns out, each of these enzymes is a protein. Right? So this is a protein, which means that it's probably has some sort of polypeptide base to it. Right? And since it has a polypeptide base to it, we would get that by translating an mRNA molecule and then get that mRNA by transcribing some sort of DNA sequence or a gene. So this is transcription, this is translation. I just did it upside down backwards here because I started with the polypeptide, right? But I get this. Of course, all of this takes energy and resources. So if I don't need tryptophan, if the cell does not need this tryptophan, instead of just, in addition to feeding back this way, I'd slow down the production of tryptophan. This tryptophan can also save time, energy, and resources by turning off the expression of any genes that make these enzymes, or that code rather for those enzymes. So tryptophan now can act as an allosteric inhibitor to some sort of enzyme to even just stop the expression of all of these genes, right? So tryptophan can allosterically inhibit what enzyme? What enzyme do you think would be responsible? What enzyme could you stop? Could you inhibit? Could you turn into an inactive enzyme to not get this gene to be transcribed, not get this gene to be transcribed, and so on and so forth? That you could stop expression of all of these genes in one fell swoop. Right? That's kind of an open question, right? So you should be able to answer that, come to class, and sort of discuss that. Okay? So just a quick little one there. Again, if you have questions about this negative feedback or that allosteric inhibition, come and talk to me about it. Ask your colleagues, your peers about it. And then, can you do this? Can you describe how negative feedback could alter the expression of genes, just kind of, again, in general, right? What enzyme would you, or enzymes would you predict could be made inactive? Okay? Thanks for listening.